So good morning, everyone. Welcome to the session. Welcome to the session of immunology and immunotechnology. And today we'll be talking about from where we left in the last lecture. We were discussing regarding the T cell receptor signaling part. Now, in that receptor signaling part, what we have established, what we had established actually till now, we had established the basic understanding of the complete receptor structure, how that T cell receptor is constituted, what are the co-receptors, what is CD3 in terms of complete T cell receptor complex, and we also understood that how different CD4 molecule and CD8 molecules are present on T cells. How many domains are there on this on these receptor molecules? So the today's lecture objective and the overall, in a nutshell, what should you be able to learn from today's lecture is single most point after understanding the receptor part, how the binding of the ligand leads to the activation of T cell and what are the signaling mediators actually that are performing that particular function. So just to start and just to bring back your memory of previous slides, I have incorporated first two slides just to brush up what we have discussed in the previous settings. Now, see, this is what we had discussed in the previous lecture, that there is an antigen presenting cell which will be processed, which will be presenting the processed antigen in the form of peptide via MHC molecule. And this particular peptide is recognized by the T cell receptor, which is alpha beta chain or gamma delta chain. Now, along with this receptor, there is a co-receptor, that is CD3, that is also required, as well as CD4 and CD8, which are specific towards MSC molecule. And this particular binding determines the specificity. And this is the picture that we discussed in detail along with the animation that we had discussed in the previous lecture. This is your T cell receptor. We had CD3 in form of gamma epsilon and delta epsilon, as well as the homodimer of zeta, which contains multiple item motifs, and single item motifs are present in the cytoplasmic tail. And we had established that T cell receptor has a very short cytoplasmic tail. So we will be pitching up from where we had left. Before that, let us discuss a particular part that last time that I asked you a single question, a very simple question, in terms that if you are provided with a mixture of a cell, mixture of multiple cells. Let's say I have T cells, I have B cells, I have macrophages, I have dendritic cells, I have dendritic cells. Now the mixture is provided to you in lab in the form of an appendoph. You have this complete mixture in the appendoph. How will you isolate T cell from this population? How will you isolate T cell from this population? That is the basic scenario. Now, see, there are multiple methods that are multiple instrumentation based methods that are utilized for this particular aim. But all those applications are based on a very single concept. So your job is to understand that concept first. Now, what is the concept? We have studied T cell receptors and the molecules that are present on the membrane. We had also studied B cell molecules. We have not studied till now macrophages and dendritic cells. Now, suppose this is a T cell. I told you that on its surface, we have a molecule called as CD3 complex. CD3 is there. We have CD4. We have CD4 that is specific for T helper cells, and we have CD8 that is specific for cytotoxic T cell. Now this is what we have established in previous lectures, and you are well versed with this information. Fine. Then we have B cells that will be expressing their own B cell receptor and the other molecules that are present on this, like CD80 and other molecules that are there. Fine. Likewise, macrophages and dendritic cells will be expressing their own set of surface receptors. Now, if I want to specifically isolate T cell, what can I use? I should use a property which is specific to T cell only. Now, what is specific for T cell? See, for T cell, the characteristic marker that is used for isolating from a population is CD3. Is CD3. I'll be using CD3 marker to isolate the complete population of T cell. Now I have multiple cell types. I have different cell types. How I can select this particular CD3? Now the answer is based on affinity and based on a single concept of affinity binding, that is anti-CD3 antibody, anti-CD3 antibody. You will be using these antibodies. Now these antibodies will be specific for CD3 only. So they will be binding to the CD3 molecule and they will never be binding to any other cell in the mixture. Okay, so what I did, I have a mixture of different cells, T cells, B cells, macrophages, and dendritic cell. I added this particular antibody inside this cell and incubated it for 2 hours. Let's say I have incubated it for 2 hours, 2 to 4 hours. You can go for incubation. Okay. Now what? It will go and bind to CD3 molecule. But then how will I separate? Now that is the point. The antibody, what we are utilizing here, the antibody, what we are utilizing here, it is modified in different terms depending upon what type of methodology you are using. Suppose I have tagged the constant region with magnetic beads. What I have done, I have tagged it with magnetic beads. Fine. I have tagged it with magnetic beads. So the complete antibody is a conjugate with a magnetic bead. Now it has bound to the CD3 molecule with the bead. Now in this mixture, I have T cell antibody complex and I have B cell, I have macrophages, I have DCs. What I did after 2 hours, I took this particular solution and placed a very high powerful magnet here, a magnet. So this magnet, what it will cause? It will cause the attraction of this magnetic field, and because of the presence of this magnetic field, all the T cells that are bound with the antibodies, all the T cells that are bound with the antibodies will be coming at the bottom due to the electromagnetic force of attraction, and the remaining cells will be present in the supernatant. So what you can do, simply, all the cells are settled here, you can simply remove the supernatant, and in the leftover with the magnet attached, you will always see a cells. Those are T cells. Those are specifically T cells. This is called as magnetic base or magnetism based antibody mediated isolation of T cell. So it's a very simple setting, but the concept is what? I'm using a specific antibody that is targeting the cell surface receptor. So this is just a glimpse of one type of methodology that is called as magnetic sorting. What I am doing, I am sorting a single type of cell from a mixture. That's why we call it as magnetic sorting in terms of immunology. Then there is another technique that is very sophisticated technique, and you people might be well versed with the technique. The technique name is flow cytometry. Flow cytometry. In fact, flow cytometry, in fact, we are calling flow cytometry just to identify the cells. But if you are using this particular instrumentation or this particular concept to purify the cell or to isolate the cell, then we call it as FACS. Then we call this as FACS. What is FACS? Flow cytometer assisted cell sorting. So what I am doing, I am using flow cytometry to isolate a single type of cell, or you can isolate multiple type of cell also with these with today's advanced instrumentation. You are isolating cells from a given population, and that particular technique is called as FACS. Now it is nothing different. There is no difference between the concept of magnetic sorting and this. Yes, you are using magnetic or magnetism. Here you will be using the hydraulic focusing based on the instrumentation. So we will be discussing the instrumentation of flow cytometry in the separate part, but let us see how things are done here. Again, I have a mixture of multiple cells. Fine. I'll be adding my antibody. I am adding my antibody. What antibody? Again, it's an anti-CD3 antibody, which is specific for T cell receptor, anti-CD3 antibody. Now, this antibody there, I had conjugated it with your magnet bead. Now here, I'm not conjugating it with my magnet bead. I'll be conjugating it with a fluorochrome. I'll be conjugating it with a fluorochrome. Now, what is fluorochrome? A compound which will be giving you the fluorescence, which will be giving you the fluorescence. So suppose I have conjugated it with green color fluorescence producing molecule that is called as FITC, fluorescein isothiocyanate. Now what is this? This is a compound that exhibits fluorescence, and you understand clearly what is fluorescence. Fluorescence is appearance of light when the electron is shifting from excited to the ground state, and that light is falling in the region of green color, in the region of green color. So what will be happening? All the complexes, this is the T cell, there is CD3 receptor, and my antibody is bound to it along with a green color molecule. So even if I give you a sample and I want you to count the CD3 cell, you will be focusing on how many green colored dots are there. And same is the concept of flow cytometry. Now, after conjugating it again for 2 to 4 hours, you put this complete mixture in form of this machine called as flow cytometer. You put the instrument here. It will take up the solution inside. It will take up this particular solution, the complete mixture inside. It will see how many cells are there that are tagged with a green color producing compound. And that complex is nothing but T cell along with conjugated antibody, and it will sort, it will keep giving you the cells which are tagged with antibody with green color fluorochrome bound to the cell, and you will be getting it in a separate collection tube. So from the complete mixture, based on the fluorochrome and based on the affinity of anti-CD3 antibody, you have managed to sort the T cell from the complete population. Fine. So these are two methods how you can go about for separating the T cells from the complete population. Now, you understood these two mechanism concept. Now, suppose if there is an interview and you are stepping inside an interview zone, and fortunately, the examiner asks you, from this population, you have understood how to isolate T cell, but I want you to isolate T helper cells. I want you to isolate T helper cells now. Then how will you do? It's very simple. You have understood the mechanism. Please try to understand the concept. Concept is using this particular marker. Now, what is the marker for T helper cell? For T helper cell, the marker is CD4. The marker is CD4, which is specific. Now, in many labs, what we do, like in our lab, what we do is first we isolate based on CD3, and from that population, you can again rerun the sample with using CD4 antibody, or directly from this mixture, you can add anti-CD4 antibody, and you will have the same result. For cytotoxic T cell, it's CD8. So what you need to understand, what you need to remember, the interviewer is asking two things: whether you know the specificity of different type of immune cells, and second thing, after knowing that specificity, whether you know how to utilize that particular concept into an application part. That is the concept, and these are the questions that do come in different settings, whether you're going for competitive exams, whether you're going for interview appearance. These are the things that you should be aware how the things are done. Now, when I say a statement that yes, T cell receptor is this, so somebody in the past must have isolated the T cell first, and from that T cell, he or she should have isolated this particular receptor from the membrane, stabilized it, and then studied it, and then a single statement was given that it contains alpha and beta chain. So you please understand how much effort is being given in the applied part to understand the single events that are happening and that we are discussing as a compiled part. So that is the thing that I wanted to discuss with you all before commencing our today's lecture, because last in the previous lecture we had discussed multiple facets of this particular T cell receptor signaling, and we left at with this particular question. So I hope this is part might be clear with you. Then let's move ahead. Let's move ahead with our story of T cell signaling. And after understanding the part of T cell signaling, that how these receptors are present on the membrane, let's talk about how they actually execute it. Now, please understand what I am trying to narrate you here. This is T cell. We have a T cell receptor. Fine. Alpha chain, beta chain. We have T cell receptors. CD3 are there, CD4 are there. Okay, it will go and bind to the antigen presented by the MSC molecule. Please listen me carefully. This binding, this signaling of and binding to the antigen receptor and binding of CD4 to the MSC is not sufficient to activate the T cell. It is not sufficient. The T cell needs extra stimulus. Now, please understand why it needs an extra stimulus. This is a T cell which is never exposed to any kind of antigen. It is the first time exposure for T cell. Those type of T cells are called as naive T cells. Naive T cells. Now these naive T cells require extra stimulatory signal along with this particular signal to get activated, and that signal is provided by another receptor, that is CD28, which we had discussed in very brief in the previous setting. CD28. Now this CD28 is present on T cell. This CD28 is present on T cell and it binds to the antigen presenting cells counterpart. Now what are the counterpart? A T cell will be having a T cell receptor, will be having a T cell receptor. It will be having CD4 or CD8. Simultaneously, there is also a presence of molecule called as CD28. Now, this CD28 will bind to the receptor present on antigen presenting cell. There is MSC molecule, there is a peptide, and that story we already know. We are talking about CD28. It binds, please understand, it binds to another receptor that is present on antigen presenting cells, and that is called as CD80 molecule. CD80. So you see there are two bindings that we had talked about. One binding is MSC along with T cell receptor. Let me phrase it clearly, T cell receptor complex. Now in that complex we had discussed T cell receptor, we had discussed CD3, we also talked about CD4 or CD8 depending upon what type of T cell is there. Second interaction that we are talking about is CD80 along with CD28 present on T cell. CD28 present on T cell. So that is the scenario that you should always remember. Once both the signals are done, then there is full activation of T cell. And in fact, when we discuss that there is a release of interleukin 2, IL-2, which will give rise to the clonal propagation, that release or synthesis of IL-2 inside the T cell receptor is mediated only and only when this particular CD28 is bound to CD80. So that is the reason why for complete activation we say that CD28 should always bind to CD80. Then let's talk about the another player. After establishing the receptor based things, let's go down now. Let's go to the cytoplasmic part. Now see here, this T cell receptor is present on the membrane. This membrane is, you know that it's a phospholipid membrane. It is rich in cholesterol. Now with this receptor in a very close proximity, suppose this is CD4, then very nearby another very important protein called as LCK is present, always associated with CD4 or CD8. Along with CD4, CD8, there will be a protein that is called as LCK. What is LCK? It's an enzymatic protein. What kind of activity it is having? It is having an activity of tyrosine kinase. It will cause the phosphorylation on tyrosine residue. So that is the reason why we call it as SRC family tyrosine kinase, called as LCK. So LCK is an enzymatic protein that is present associated with the CD4 and CD8 molecules that are present at the receptor or the proceptor. So the complete cluster is present. Now what will be happening? Once the antigen binding has taken place, this particular LCK now comes into very close proximity, with the complete complex. It comes into very close proximity. Now, once it is coming to the close proximity, what is the job of this LCK? Job is very simple. The CD3 molecules I told you contains different numbers of items. Items are what? Items are the motifs of tyrosine residues. Now these residues will be getting phosphorylated. These residues will be getting phosphorylated. Now this particular function of attaching phosphate residue on the item motifs of CD3 receptor is the function of LCK. So till now what is happening? This is a T cell receptor, this is a membrane, this is a T cell receptor, this is bound to the MSC molecule, CD4 is there, and along with that CD3 co-receptor is there. Now this is complete TCR complex. Now in this CD3 there are item motifs. There are multiple item motifs because of the long cytoplasmic tail. Now another set of CD28 has bound to this 28 is bound to CD80. Now that story is done. The moment this binding is achieved, there is another protein called as LCK that is present. Now let me recorrect myself. There is a LCK that is present in this fashion. There are multiple CD3 molecules that are present here. There is again T cell receptor complex that is present here. Once this is bind to the antigen, this LCK will come near to this CD3 and will cause the phosphorylation on the cytoplasmic tail region on the motifs called as item motif. This phosphorylation, addition of phosphate group is very much crucial in order to activate the signaling. Now what is happening? See, this is the exact representation what I want to tell you. There is a T cell receptor, processed antigen, MSC molecule bound. Then there is a co-stimulated CD4 which is bound to the MSC, and we have a complex of CD3. These three molecules combine to give 1, 2, and 3. These three are collectively called as TCR complex, T cell receptor complex bind. So once this binding has taken place for all the two, then there is a specific set of protein called as LCK, which is tyrosine kinase. It will get activated and will phosphorylate the tyrosine residues that are present on the item motifs. These are item motifs. Now see, they are getting phosphorylated. Now next is what? I told you in B cell that if a protein is getting phosphorylated and another protein is coming and binding to this particular region, then this portion that I am highlighting is called as SH2 domain. And all the proteins that are having this particular SH2 domain will be able to interact with the phosphoproteins in a lock and key manner. Then what is happening next? Once these items are phosphorylated, next there is another protein called as ZAP70, which is having the SH2 domain here, will come and bind to all the phosphorylated residue, Once this ZAP70 is bound, now who will tell ZAP70 that please come and bind? That is again the job of LCK. It will signal ZAP70 that I have phosphorylated the item residue. Please go and bind to this particular to this particular ITAC motif. And you see here, that is what I have shown, that LCK also phosphorylates ZAP70 and thereby it activates ZAP70. Thereby it activates ZAP70. Once this particular ZAP70 is activated, once this ZAP70 is activated, then the downstream signaling starts. Then the downstream signaling starts, and the signaling which was happening in B cell is common with what is happening in T cell. That what is happening in T cell. Now see here, see this particular panel for your reference. Once the ZAP70 is bound, then it will activate another adapter molecule called as linker protein of activated T cell, or LAT, linker protein of activated T cell, that is always present attached to the plasma membrane. This ZAP protein will cause the activation of LAT molecule that is present with the membrane. Now, when I say activation, I mean it will induce the phosphorylation of that particular molecule. It will induce the phosphorylation, and this is how the complete narration is. ZAP7 is activated. It has now activated the LAT. There are multiple phosphate residues that are attached to this LAT. And now this particular LAT protein will serve as the platform where multiple people can come and stand. Where multiple people can come and stand. And all the immunoproteins that are having this particular SH2 domain will now be coming and will be attaching themselves to the LAT protein, because it's a very big protein. It has the space to accommodate multiple players, and such player is your phospholipase C gamma. One such player is your phospholipase C gamma. Now this phospholipase C gamma, what I'm highlighting, don't worry about this part. We'll be studying in detail in the next session. The phospholipase C gamma, which is getting activated. How it is getting activated? It is going and attaching to the LAT, which is in phosphorylated state, because this PLC gamma also contains a SH2 domain, which is specific for phosphate residue. Once this PLC gamma is activated, it's very simple now. It will act, it will cause its function. It will phospholipase. It will break the lipid in phospholipids. Now, how it is breaking? It will lead to the formation of IP3 and diacylglycerol, DAG. This IP3 will be responsible for increasing the intercellular concentration, whereas diacylglycerol will be responsible for activating protein kinase C and the other MAP kinases. And collectively, they will lead to the effector function of immune cell. They will increase the T cell proliferation. They will increase the T cell effector function, and they will increase the synthesis of IL-2 cytokine. And this IL-2 cytokine will be released, and then it will signal the other T cells to divide and clonally propagate to increase their number. And this is how the complete signaling is achieved. This is how the complete signaling of T cell is achieved. Now, apart from this, see, biologically, any cell cannot rely on a single pathway. Suppose somehow, if this single pathway is affected by a pathogen, then the complete immune response will be diminished. So our immune system is very intelligent. It is not relying on a single type of cell. No, it is not relying on a single type of cell or signaling molecule. It has other associated molecules also that are going on a separate line but performing the same function. Like what? Like there is another adapter protein present here, which will lead to the activation of RAS, and it will lead to the activation of this particular axis. So there are multiple axes that are present along with this particular LAT. That is why LAT, because of its bigger size, can accommodate multiple players which can perform different functions. So you see, this is how the complete signaling is going about. Now, when I'm saying about PLC phospholipase 1, let us see how it is mediating the function. It's easier said than done. So we have phospholipase C gamma 1. It's an enzyme, lipase, Fine. Is an enzyme. It will be having its substrate. The substrate is PIP2. PIP2. What is PIP? It's a phospholipid. It's a phospholipid made up of inositol phosphates and diacylglycerol. It will break down its constituent into its constituent component. One is IP3, that is inositol triphosphate, and another molecule is diacylglycerol. See, lipids are made up of like this, a fatty acid, how it is made up of glycerol along with an acid. So we have inositol and we have diacylglycerol. This inositol, this IP3 which is released, it leads to the activation of release increase in calcium concentration in the cytoplasm. Increase of calcium concentration will lead to the activation of its dependent protein called as calcineurin. Calcineurin. Now this calcineurin, what I am saying, calcineurin is the protein exactly which is getting activated in response to the increased calcium ion concentration inside the cytoplasm. Once this particular calcineurin is activated, it signals, it activates a transcription factor called as NF80, nuclear factor of activated T cell, NF80. This will get activated and then it will migrate from cytoplasm to nucleus, and nucleus it will go and bind to the promoter region because it's a transcription factor. It will go and bind to the promoter region, and here it will lead to the expression of the genes which are present downstream, and these genes are nothing but IL-2 synthesis gene, which will be synthesizing IL-2, the genes which are responsible for cell cycle progression, cell cycle proliferation, or pro-inflammatory cytokines that are required. So that is how the complete pathway is mediated. Not only that, this particular this is the axis what we have discussed for a single molecule that is produced, that is IP3. This axis belongs to IP3. Now, what about diacylglycerol? What about diacylglycerol? See, diacylglycerol will go, this diacylglycerol which is formed will go and activate another enzyme called as protein kinase C. Now, protein kinase is phosphorylating capability. Protein, the substrate will be protein. It will lead, it will lead to the subsequent path. It will lead to the activation or phosphorylation of NF-kappa B. Now, this NF-kappa B is a heterodimer made up of P65 units and P50, and it is inactivated because of its inhibitor, I-kappa B alpha. So it will go and phosphorylate this particular inhibitor, and it will lead to the activation of NF-kappa B, and then it will go inside the nucleus. This single unit of P65 and P50 will be going inside the cell. They will binding to the promoter, and then again they will cause the expression of downstream part. And these are nothing but all the pro-inflammatory cytokines that you are talking about. The inflammatory response that is required to generate all the proteins that are responsible for generating those information will be getting activated and synthesized. So this is how T cell is actually giving rise to the complete response by dividing it into two different signaling cascades. One is mediated by IP3 and NF80. Another one is mediated by diacylglycerol and NF-kappa and NF-kappa B. And this is how the complete process activates. The diacylglycerol has activated PKC theta, protein kinase C theta. It will lead to the subsequent events leading to the activation of NF-kappa B, which will go inside the gene and will cause its activation. Now, this complete signaling pathway is very important. We in lab we can establish the complete pathway. I can see whether this is involved or getting activated. So these names, Karma1, BCL10, MALT1, TRAF, Nemo complex, I-kappa B alpha, all these pathways are made once we have experimentally validated each step inside the lab. And we regularly do this in the laboratory settings. But for you, you should clearly understand that once the protein kinase C theta is getting activated, then there are multiple pathways through which multiple signaling molecules which are leading to the single effector function that is getting NF kappa B activated. Now, for activating it, it has to degrade its inhibitor, and then NF kappa B will move inside the nucleus. Nuclear translocation will be there, and then it will lead to the binding to its particular promoter region, leading to the activation of the genes which are actually involved in inflammatory responses. That is how the complete signaling is going about. So I hope in today's session you had a very clear cut understanding of how the things are moving, how things are actually going, and how when we are talking about that a T cell receptor is there, then how it mediates its complete function. So in next lecture, we will be talking in detail about how this T cell receptors diversity is maintained based on genetic level, the genetic recombinations we'll be talking about. So these are the MCQs that are required or that will be assisting you for brushing up what we have discussed till now. So thank you so much for your patience listening, and I hope that the lecture was fruitful to you. And please again go through the lecture in order to have a better understanding of what we have discussed till now. So till then, stay safe and wish you a very happy learning.